Ramakrishnan, shall I start? Shall I start? Can you see my slide? Can you see my slide, or once again I should share?
Yeah, it is visible.
It's visible now. Okay, so sorry. Okay, then I'll go.
Yes, please.
Okay then. So anyway, I won't take much time. I'm going to in this session I'm going to cover the various various properties about the tobacco mosaic virus. So I don't know how many people, like maybe you would have read in the textbook even during the school days also. So I would like to cover the so we always work on the molecular biology aspect of the viruses. So I would like to cover in that way also. So if you have any doubt, you can stop in between also. It's a very good, I will, after reading so many papers about the tobacco mosaic for the lecture, I got interested on tobacco, doing research on tobacco mosaic virus. So that is, so it made me more interesting on about the tobacco mosaic virus. So I know how you're going to be more interested after my lecture also about this tobacco mosaic virus. So if you see that one, tobacco mosaic virus, it is more important in the case of the plant viruses. In the sense, this is one of the very old virus, like this was one of the virus. Not only one, this is the first virus was identified century ago. So it is considered as a very old plant viruses also. So if you see that one, these viruses from the name, you can understand tobacco mosaic virus. So this particular virus infect tobacco plants. Not only that, it also infect the other members from the Solanaceae. So you are from basically our botany students, you know what other plants also comes under Solanaceae, like tomato, brinjal, and other so many plants also. So this was actually in 1886. The scientist, the guy Adolf Meyer, he observed the tobacco, the symptoms on the tobacco plants. So that time he didn't know that the symptom could be due to only the tobacco mosaic virus. And in the beginning of my session, I discussed various developments happen on the viruses. So during that time I mentioned about this Beijing. So he tried to identify what is the thing is responsible for the infection by passing the extract of the tobacco mosaic diseased plant, and based on his study, he concluded that this is a new way of infectious agent, and the agent could be the acellular also. Okay, so for the people know only about the bacteria, so he made a conclusion that it could be some micro which cannot be seen under the microscope, cannot be a cellular also. So later this vendor Stanley, he also got the Nobel Prize in 1946 for purifying this particular virus. So that time he made the thing that it's a particle nature. It is basically made up of protein. But later this Bowden and then Perry, in the same year, they demonstrated that. So they by doing experiment, they proved that one tobacco mosaic virus, it's not basically a protein, it is also contained the nucleic acid. And then mid-50s, the scientists from Germany and then States, they made an infection by using the viral RNA alone. Okay, that is a very, very important discovery. By which the people they started to make the infection by using the just alone nucleic acid. So that was one of the great discovery happened in 1950. So these are the different developments happen in the case of the tobacco mosaic virus. So if you take whoever worked on the virus, that means a plant virologist, they start their career from the tobacco mosaic virus. So then only they will start moving to the other viruses. So that this tobacco mosaic virus gave the basic platform for the many virologists also. So to tell more about the tobacco mosaic virus, it is known for several firsts in virology itself. If you see that one in the virology field, for so many reasons, tobacco mosaic virus was considered as the first. So in the sense, this was the first virus to be purified. And also, this was in this particular virus, they proved that viruses consist of RNA and then protein. And also the first X-ray, the first crystallization was done by with tobacco mosaic virus. The first viral thing was crystallized with tobacco mosaic virus, and also through the extracrystallographic diffraction thing, they also showed the helical structure. That is also first for in the case of the virus, and also for the first virus to be seen under the electron microscope, and also for to know the sediment velocity of the virus by using the analytical ultracentrification. For that purpose also, this is the only virus, otherwise the first virus to be used to determine the sediment velocity also. And also the first the complete genome of the virus also available was sequenced for the tobacco from the virus side. It was from the tobacco mosaic virus only. And also the previous session, I told what are the different ways we can make the viral resistance plan by the concept called a pathogen derived resistance. For the first time, for the plant virus, more specific tobacco mosaic virus, by using the core protein, they showed that it showed the resistance against the tobacco mosaic virus also. And also the people they identified there is a resistant gene which is responsible for resistance against the tobacco mosaic virus, also identified in the tobacco plant. For all these purposes, for all these things, they happen first time with the tobacco mosaic virus only. So it holds as an overall topic in the virology field also. So don't think that still a lot of things are coming up. So even these viruses are also useful to display the epitope for any particular other pathogens also. So thing is that you can display the epitope on the core protein of the several viruses, and this tobacco mosaic virus is highly useful. Not only that, it also has a lot of other applications in the nanoscience also. So that also, in that aspect also, tobacco mosaic virus is highly useful than any other viruses. I have papers. I read that one. Really, that time only I also came to know that a lot of research has been going on the tobacco mosaic even now also. So with this one, I would like to get into that. What are the structures, what are the components, and how the infections happen. So that happened that I will be dealing in my talk also. Okay. So if you see that one, as I told you, it doesn't not only infect the tobacco plant, it will infect all other plants, comes under the Solanaceae plants also. So whenever it infects in the other plants, it shows the other symptoms also, from the mosaic, mottling, necrosis. Not only that, even the tomato plant, it infects the tomato fruit itself. So that means it causes the broad spectrum of symptoms on the different plant in the different part of the same plant also. Okay, so coming to the morphology, the structure. So as I told you, it has a it is basically a rod-shaped structure which has a helical symmetry. So this virus comes under the genus called as Tobamoviruses. The family is Virgoviridae. So this guy is the Franklin one. He identified described the structure of the tobacco mosaic virus on the first time. So if you see that one, it has a 4 nanometer hollow tube inside, and then it has a size of the 300 nanometer into 15 nanometer size of the thing. So if you see that one, it has a RNA molecule, viral RNA, which is covered by the capsid with the number of 2,130 copies of the code protein. The size of the RNA is 6,395 nucleotides. So all the tobacco mosaic has a viral RNA has a size of 6,395 nucleotides. So if you see that one, each capsid is made up of 115 amino acid. Each amino acid, the code protein, the capsomere, the size of each capsomere is 17.5 kilodalton. So this is a electronic. This is a structure got it through the electron microscope. So To see more further that one, so each turn, so this is one turn, so each turn as a is covered by the 16.33 capsid subunits. So you can count the 1, 2, 3, 4. If you count that one, so each turn of the RNA is surrounded by the 16.3 subunits of the capsid protein. So if you see that on each subunit covers the 3 nucleotides, you can see it here. So this picture clearly tells that one. Each subunit it is covered by it covers the 3 nucleotides. So as together, each turn has a 49 nucleotides. The 49 nucleotides. So the 49 nucleotides is covered by the 16.33 capsid subunits. Okay, so this is regarding the structure of it. So as a central axis, you have the RNA, and that is covered by this dot shape, this covered by the capsid protein also. So coming into the genome, so genome size is the exact size is 6,395 nucleotides. So it is a positive. It has a positive single-stranded RNA. So the positive, what I mean is that, so this RNA, it is very viral RNA is very similar to the mRNA of the host. So what happened as soon as the virus got into the host plant, then there will the disassembly of the capsid happen, and then the RNA can directly go for the translation. The protein will be produced, and then it will, and then subsequently the life cycle of the tobacco mosaic will be continued. So if you see that one, as I told you, it's the single-stranded RNA, persistence RNA, and the 5' it has a cap. So this is a cap. So as you all know, mRNA of the plant mRNA, they have a G cap at the 5' end. Similarly, this also has a G cap at the 5' end. On the 3' end, it has a tRNA-like structure that is very useful for during the replication function. And it also in the 5' end and the 3' end, they have a UTR, that's untranslated region. So in the untranslated region, it has a sequence that is responsible for binding the ribosome subunit. So they call us omega subunit. That is called as omega. The region which is responsible for the translation thing, they call as omega sequence. So they have a specific sequence in which ribosome binds, and then they go for the translation. And if you see that one, it's very, very interesting is that it codes only the 4 proteins. One is replicase. In the replicase, there are 2 subunits. One is 126 kilodalton, and the other one is 183 kilodalton. And then the other 2 protein called as movement protein and then core protein. By this 4 protein, it will bypass all the thing happen in the host cell, and it uses the host machinery for its own replication and then translation also. So really it is very interesting to see that one. So the minimum with the 4 protein, so by having the interaction with the many number of protein, they can execute the infection in the host plant itself. So thing is that, so from this sense strand, that means from the viral RNA strand itself, directly the replicase protein will be produced. But the other 2 protein, movement protein and then core protein, they are produced from the subgenomic RNA. I will tell you what is a subgenomic RNA. In the sense, what happened, the translation it won't happen in the viral strand. So what happened, from the positive strand from this region, the negative strand will be produced. Okay, the negative strand will be produced, and further this negative strand will be connected to the positive strand, and that from that positive strand, the positive strand will be coded for this movement protein and then core protein. So what happened, the positive strand, whatever produced from this parent strand, there are 2 different subgenomic RNA will be produced. From each one particular protein will be produced. For example, from this particular thing, the movement protein will be produced. From the other subgenomic RNA that is produced from the 3' end of the viral RNA. So that will produce that will code for the capsid protein. Okay, so this is the size of it. Moon protein is 13 kilodalton, core protein is 17.5 kilodalton, and the replicase, I told you there are two subunits. One is 126 kilodalton, the other one is 183 kilodalton. I told you there are two subunits the replicase protein. There are two type of things. One is 126 kilodalton and 183 kilodalton. So what happened there? Ribosomes. Okay, any that I will explain when you go for the protein synthesis. So time being, I'll stop discussing about the genome here. So what happened if you see the function of the each one? As I told you, there are in replicase two things. One is 126A and 183 kilodalton. The 126 has a two motif. Both these protein, they are involved in the replication of the viral RNA. Among these two protein, the 126 is a small protein when you compare with the 183 kilodalton. So what happened? It has a two function. One has a methyl transferase, the other one is a helicase function. So those two functions are necessary for the proper replication of the viral RNA. So that will be governed by the 126 kilodalton protein. The other 183 kilodalton protein, which is a real RNA dependent, which is a real replicase protein, which has a motif for the RNA dependent RNA polymerase that execute the real replication of the viral RNA. And this during the virus infection, there is one process happen that is called the silencing. This I explained during the RNA mediated protection. So the silencing is basically RNA based system. So whenever the system whenever it sees the double-stranded RNA formation in the plant, so what happened? The silencing mechanism will be executed. Similarly, during the replication of the tobacco mosaic virus, the silencing mechanism will be evolved. So that is to defend against the virus. To have a counter defense, what happened? The protein, the 126 kilodalton protein, which has the activity of the suppressor of gene silencing. So what happened? That will interrupt or it will disrupt the silencing mechanism by which it has a counter defense mechanism, and through which tobacco mosaic survey, and then it will go for the further infection. Okay, so this is the function of these two protein. Coming to the 30 kilodalton protein, that is a movement protein. The movement protein has many functions. More specifically, it will helpful to transfer the virion from the infected cell into the new cell. So that means it is very important for the cell-to-cell movement of the virus. And then core protein has several functions, and its size is 17.5 kilodalton. It is also responsible for the long distance movement. So after it moves from one cell to another cell through the plasmodesmata, finally it reaches a different part of the cells through the phloem. That is happened with the help of the core protein. Apart from it has a protective role for the nucleic acid, it also helpful in the cell-to-cell movement, and also in the long distance movement through the phloem also. Okay, if you see the now we'll see the life cycle or disease cycle and epidemiology. The first I will deal with the disease cycle. So here the transmission, generally viruses are transmitted, more specifically plant viruses are transmitted by the vectors, by the insect. More often it is transmitted by the insect. But in the case of the tobacco mosaic, I really surprised that very rarely it is being transmitted by the virus, transmitted by the vectors, that means insect. Most of them, what happened that it will go through the stomatal opening, or in some cases what happened, it goes through the wounding itself. So when there is a contamination of the virus from the infected plant, when the two plants rub each other, from the infected plants through the healthy plant, it is traveled through the wound tissues. In some cases, the people they observe that even it goes through the stomata also. And some cases what happen when you use the contaminated tools or through the contaminated worker hands also, it carried into the new plant, and that causes the symptoms also. So the people also observe that it also been transmitted by the seeds, pollen grains, and very rarely happen with the insect also. And during the wind or during the water, like the water movement through the water also, people they found that the virus is being transmitted. And if you see that one, it is a tobacco mosaic is extraordinarily stable in the sense even if you store it at 4 degrees Celsius in a fridge or in a freezer, it is alive for 50 years. The people they confirm it by keeping it for 50 years, and then after they check for the activity, even after 50 years, it shows the symptoms on the inoculated leaf also. Okay, so if you see that one, there are different time point in the infection. That's called as earlier infection, intermediate, and then late infection. So this whole life cycle is being depicted in this picture. So tobacco mosaic virus comes into the
the slide. It's not in the full view.
Sorry. No one told me that. Okay.
No. So far you have mentioned only now. We have
text.
To see.
Okay, now you have to see the pictures now. Okay. Sorry.
Yeah.
Thanks, Govt. Thanks, Govt. So what happened? So this tobacco mosaic virus comes into the host cell and reaches the cytoplasm, where the disassembly happens. The disassembly also not a complete process.
Still, it's not in the full view.
Dr. Gopal, it is in full view only. Now only.
Okay,
Right.
Okay, fine.
My
okay.
mistake.
Okay, sir. So after the tobacco mosaic virus reaches the cytoplasm, disassembly happens. I will explain how it happens there in the subsequent slides. So what happened? The disassembly is not a complete thing. Only it happens at the 5' end. So during this process, translation happen, and then it will produce the replicase proteins alone, and then the replicase proteins goes to the 3' end of the viral RNA, and then the replication happen. So, during generally the replication happen in the endoplasmic reticulum, and what happened? Meanwhile, the movement protein also produced. So that will take the whole virus into the plasmadesmata, and then it reaches the next cell. So after that, it reaches the phloem, and then to the different part of the plant also. So I will go through one by one. So basically, this is how the life cycle includes the different stages. One is entry into the host cell, uncoating, biosynthesis, assembly, and then release. Coming into the entry, this I clearly told that through the wound tissues, it get into the new cell, and then the uncoating. The next step is the uncoating or disassembly. So what happens is that the people they identify the carboxyl amino, they're like aspartic acid, the glutamic acid. They have a COOH group. So different acidic amino acids, they interact through the carboxylate interaction. So that is very, very important for the intact structure of the capsid. So and then there is a interactions also happen between the same carboxylated amino acids with the phosphate group present in the viral RNA also. So what happened? So the people, they even identify what are the amino acids which is responsible for this interaction. So when the virus reaches the host cell, the pH is different. The pH is alkali. So what happened? And also there is a limitation of the calcium in that also. Whatever the interaction I told, that for that interaction it always needs the calcium. So as I told you, the COO minus basically acidic. So two acidic cannot interact. So for that it needs the calcium Ca2 plus ion. So what happened? And also it also pH dependent. So when the virus reaches the host cell, it has a different pH, the higher pH, the alkali pH, and also the system lacks the calcium that is very much needed for the carboxylate interaction, and subsequently it is very important for the intact capsid structure. So once it reaches there, due to this higher pH and the deprivation of the calcium ion, so the disassembly begins. That means the interaction between the two carboxylate amino acids started to the bond breaking happened there, and that will dismantle the few amino acids. Even the people, they identify whatever the proteins, nearly something like a 23 protein subunits which covers the 69 nucleotides from the 5' end of the RNA started to dismantle. So and also the interactions between the coat protein as well as the RNA also disturbed due to the same reason, and so that now the 5' end of the RNA is exposed for the subsequent process. So what happened that so as you expect that there won't be a complete disassembly happen in the case of the tobacco mosaic virus. This is what happened for other viruses also. So this virus is so clever that it always disassembly begin only on the 5' end of it, not the complete viruses. So whatever the region exposed, that subsequently will be taken for the translation. So the people, they told that this the code translation disassembly happened. This is a term I want to introduce here, that so the disassembly is also parallelly the translation is also happened there. So that's what this process is called as code translation disassembly happen. So once the translation once the disassembly happens on the 5' end of it, the translation process begins. So as I told you, the 5' end of the RNA, especially the 5' UTR, there is a region called as omega region. So that has a sequence which is helpful to binding of the eukaryotic initiation factor 4A. So this is a thing that is needed for the ribosome binding. Especially the small ribosome, small subunit of ribosomes needs to bind there. So what happened? The 5' UTR, in the omega sequence, it has a binding site for the eukaryotic initiation factor. So the eukaryotic initiation factors, after disassembly, the eukaryotic initiation factor 4 bind to that omega sequence. That will pull that first small ribosome unit and then the largest subunit. So what happened during this process? The two replicase protein, one has a size of the 183 kilodalton, the other one has a 126 kilodalton protein are produced. If you see that one, if you see the sequence, there is a stop codon, and what happened? The stop codon is not a strong stop codon. They have the amber stop codon. So what happened? The ribosomes sometimes always slip through or always it passes through that stop codon, and due to that you will have a bigger size of the replicase that is called as 183 kilodalton. At the same time, when the ribosomes stall at that particular translation stop site, it will produce as the 126 kilodalton also. So you may ask me in what condition it will produce. Still they don't know. But thing is that by doing the various experiments, I have gone through all the paper. By doing the mutation and other further studies, the people, they claim that these two proteins are very, very important to have a very high efficient replication for the replication and for the translation also. So these two proteins are very, very important. Already I told you the function of these two proteins. 126 kilodalton, which is responsible for the methyl transferase and the helicase, and 183 kilodalton, which is really RNA-dependent RNA polymerase, that is responsible for the replication. So what happened? These two proteins are synthesized first. So as I told you, these two proteins will move into the 3' end of the viral RNA. In the 3' end, it has a structure called as tRNA. So it has the tRNA, you know that it has always have the clover leaf-like structure. So that is very, very suitable for this RNA, this replicase to bind the 3' end of the RNA. And then during this process, this assembly also happens at the 3' end. And that makes a replication. And during this process, the other negative strand will be produced, and subsequently subgenomic RNA also will be produced. So here, like this is what the biosynthesis things happen at the 5' end at beginning, and later at the 3' end after going for the replication. Okay, so the translation process is not complete. In the case of the tobacco mosaic virus, the partial translation happened at the 5' end. That leads to the replication of the 3' end, from which the other two proteins are produced, movement protein and then core protein are produced. So they will execute the downstream process only. Okay, so this is the biosynthesis steps. The biosynthesis, two things happen. One is the formation of protein, that is called as translation. The other is the replication. So I will explain the replication. So as I told you, the translation happened at the 5' end. The two proteins are produced, so they come at the 3' end. Then the subsequently assembly also happen in the 3' end. Then the first thing, they produce a negative strand RNA from the positive strand RNA of the tobacco mosaic viral RNA. So the negative strand RNA form. So when you find this positive and the negative strands, they are called as replicative form. Okay, when you see the two type of the thing, So they form the two, they call as a replicating form. So as I told you, now it looks like a double-strand RNA. So during this process, the silencing process will be, the silencing mechanism will be executed, but this 126 called is act like a suppressor of gene silencing that will disturb that process. So whatever the viral RNA, the negative strand form, from that, the another positive strand will be produced as a subgenomic RNA. So that two different subgenomic RNA will be produced, one for the core protein, other for the movement protein, and subsequently this will be taken for the translation, and then the proteins will be produced. So if you see that one, only the subgenomic RNA, which is for the core protein alone, has a 5' cap, and this one doesn't have, but still these two RNAs are stable, and they produces the protein also. And similarly, they found that several multiple negative strand RNA also will be produced. From the negative strands RNA, the positive strands will be produced. The one positive strand, the complete positive strand will be taken for the encapsulation. The other positive strand, the two different positive strands as a subgenomic RNA will be produced. So that will be taken for the translation to produce a movement protein, and then core protein also. So whatever the replication happen, this replication happen in the endoplasmic reticulum. So this is you can see the slide here. So in my cursor also. So this is a nucleus. From the nucleus, endoplasmic projection happened there. So what happened that one? The virus replication happen as a virus replication complex. So that means it forms like a factories. It modifies the endoplasmic reticulum to have a replication function in the endoplasmic reticulum. That means whatever the other protein needed for the replication will be grabbed by the viral protein, and they execute the replication and then the translation. So what happens once the translation happens, all the viral proteins are produced. So that viral proteins will form the nucleo as a nucleoprotein. So what happens then? Whatever the movement protein produced there, so that will taken this virion into the plasmodesmata, and then it moves to the next cell also. So this is how the replication happen. After the replication and the translation happen, now the virus have enough viral RNA as well as the viral protein also. Then it will start moving into the next cell also. So here for the next cell movement, the movement protein is very, very important. So here, as the previous case, what I told you that the core translational disassembly happen. Similarly, the replication along with the movement, along with the replication, the movement also happen. So that is also parallelly happened that. So whatever the replication happen here, after the replication, whatever the protein present there, that will form the nucleoprotein. That means viral, that will form as a virus. So that will be taken by the movement protein and moves into the next cell. So the very interesting thing is that so whatever the plasmodesmata size, it's very small, through which the virus cannot move. Okay, the virus cannot move. So the size of the plasma needs to be excluded. That means the pore size of the plasmodesmata needs to be increased. So what happened? The movement protein, first it interact with the cytokeratin proteins like actin and myosin. So what happened? It will bind to those proteins, and it take the whole complex and reaches the plasmodesmata, and then it will increase the size. Otherwise, what we call as size exclusion limit. So what happened? That will the movement protein binding with several host protein, that will increase the size of the plasmodesmata, and that makes movement of the virion into the another cell. Once after reaches the another cell, the same process, once again the cyclic process happen, the disassembly happen there, and then translation happen, and the further replication and further translation, it will multiply in the next cell also. So this is the movement happen. This is what the cell to cell movement happen. So if you see that one, the movement protein has a several function. It will helpful. It will bind to the very virus, and then it interact with the cytoskelet element to facilitate transport of the P30. That means movement protein, the tobacco mosaic virus RNA complex from the cell cytoplasm to the plasmodesmata. After reaching the plasmodesmata, it will increase the size of the pore size of the plasmodesmata, and also during this process, it will also associate with some other receptor protein that will be helpful for the movement also. So far the many host protein, they also notice that this movement protein, it interacted with this many proteins, like a cell wall associated protein, pectin kinase, and then movement protein, binding protein, plasmid phosphonate kinase, caliractin, and then actin filaments and with myosins, and then NCAPP1. These are all host protein. So by interacting with these many proteins, it executes the movement of the virus from the one cell into the another cell happen. And next step in the life cycle is here. You can see the assembly. So in the assembly, it generally refers how the encapsulation happened. So what happened after the replication and the translation happened in that one? The viral caps protein, that will capsid protein, it will recognize its own viral RNA, and then assembly begins. So it is so they found that it is an organized process. So what happened that generally the assembly starts from the 3' end of the RNA. By doing the several experiments, I have gone through all the papers, and they done so many experiments with that one, and they identified that the encapsulation, it begins from the 3' end of the viral RNA. So there is a special sequence are found in the 3' end of the viral RNA that shows a signal to capsid protein recognize and to start the encapsulation also. So this is how it happened. So when it produces a single subunit, so it binds to the another subunit and such a way that it will form the 2 discs. It forms a 2 discs. So when these 2 discs form during this process, it will start to. So what happened when it forms a thing? It will recognize a viral RNA as a hello. Okay, so it will recognize the viral RNA, and then after recognizing the viral RNA, slowly the disc length or disc number of the disc covers the RNA also will be increased, and ultimately all the viral capsids covers the viral RNA, and this is how the maturation happened. Now the virus is ready for the another infection also. So once it forms a virus, as I told you, through the plasmodesmata, it will go into the next cell. And during this process, if some infection happened, that means when the infected leaf, when they rub the healthy leaf through the wounded tissues, it can also reach the other part of the another plant also, another healthy plant also. So if you see the epidemiology, so it causes the disease through the season. So what happened? So we have to what happened when you do the crop rotation? When you do the crop rotation, if you use the same, like as I told you, the tobacco mosaic virus can infect many number of plants. So in the crop protection, when you use this, the plants which also infected by the tobacco mosaic virus, in that case, what happened is you cannot remove the virus from the thing. So while selecting the plants, for the crop protection, you have to be very careful that particular plant should not be infected by the tobacco mosaic virus also. So if you see that there are several ways the people they try to control this disease. In the case of the greenhouse, so they found a different way. By treating the equipments, whatever using it for cutting or something, that should be immersed in a soap or like washed with the soap. All the tools should be washed with the soap or with the 10% solution of the bleach also. And if you found that there is an infection there, you have to try to burn the plant, and then you have to try to remove that particular soil. As I told you, it is so active even for a longer time. So it is better to, if you remove the soil, it will be useful for the subsequent plantation also. And there is one mechanism called as cross protection. What is the cross protection? So what happened? There are some mild strains are also available that doesn't produce any symptoms also. So what you have to do? Initially, you have to inoculate, or you have to spray those less infective strain. So during this process, the plant will develop a defense mechanism. So when the real vigorous strain or the severe infecting strain, if it inoculate in a subsequent infection happen, still since it has already defense mechanism, that will protect from the severe strain also. So this is one way. In some cases, what happened by doing the transgenic. that means you can develop the cultivars for the cultivar, like you can develop the particular thing by breeding with the naturally resistant thing. So what happened, there is a gene called as N gene, so which is basically interact with the protein called interact with the domain which is present on the 126 kilodalton replicase protein. So during these interactions, what happened, there is a real protection happen. There is some resistance mechanism will be hello. Okay, so the resistance mechanism will be developed, so that has a resistance against the particular virus also. So some varieties, they have this N gene, more specifically tobacco plants. So when you do the crossing with those plants, by which you can have the resistance also. Otherwise, you can do through the biotechnology. You can make the transitioning with the core protein or movement protein or replication, what I told in the previous section, by which you can have the transgenic resistance plant also. So this is the one. In people, they also identify, like they have successfully, they eliminated the thing by dipping the hands and whatever the equipments in the milk, and after they went for the planting or other purpose, they found that there is no infection at all. So this photo I took it from one of the site. They showed that one, whatever the tools or hands dip in milk, they show the higher growth without any infection also. So and then in some cases, what happened, seed, as I told you, sometimes seed, it also transmits seed also. So when you treat the seed with the 10% solution of the trisodium phosphate, or sometimes we're keeping at 70 degrees Celsius for 2 to 4 days prior to planting, that also will avoid the virus infection also. But sometimes what happens, the germination will be affected. So, but you will get the virus-free plant also. So by doing all these things, you can avoid the virus infection on the tobacco or any other plant also. So I will stop here. If you have any doubt, you can ask me. Hello, students. Hello, participants. Sorry, hello, participants. Any doubt?
Dear participants, you can unmute your mic and ask
any questions if you have.
Hello. So
hello?
this is Vijay again.
Okay, ask me, sir.
Okay. He is my colleague.
Okay, I understood. In the previous section itself, while asking questions, he told clearly. Ask me, sir.
Yes, sir. Sir, coming to TMV, you are saying it is infecting tomato.
Yes,
so for tomato, there is other virus also affecting the tomato.
yes,
So
sir,
when compared to TMB, what is the native virus affecting tomato is more prevalent, or TMB is affecting more commonly in tomato plants?
No, sir. Actually, I cannot give the exact data, but it is being affected by a lot of viruses, like a tomato spotted wilt virus, and even one of the Gemini virus, like tomato leaf curl viruses, tomato yellow leaf curl viruses. So this tomato plants is being affected by a lot of viruses also. But symptom wise, as for my experiments, I will claim that this tobacco mosaic virus is not that much of effect it causes on the tomato plant when you compare with any other viruses also. So it's not that much. Okay.
Okay. Yeah, understood. So one more question I want to ask. In the TMB affecting Nicotiana species.
Okay.
Yeah. So there are several species available for Nicotiana. For example, Nicotiana benthamia, Nicotiana tabacum, and different species are available.
Yes.
So this virus affecting which species more commonly?
That data, actually, I didn't read that in that aspect, but I don't know. I can't give the answer right now.
Okay, no problem.
But the thing is that it cannot affect the plants which has like gene resistance. That is the gene responsible for the resistance. But it doesn't like it. It's not like that. Only the particular species have that gene. In all the varieties can have, but in some of the things may not have that one. But I have to look that one. Sorry, sir. I don't answer for your question.
No problem, sir.
Okay.
So what I understood is this will affect mostly Solanaceae members.
It's true. It's true only, sir. This is true.
Okay.
I will consider that's basically based on the engine only. So depending upon the similarity present there, so most of the Solanaceae family, they may have a homology with this engine. So depending upon the engine thing, so the resistance basically with respect to the engine only. So that is the one which recognize the this virus, and then in the subsequent process, it will show the resistance against this tobacco mosaic virus.
Okay, sir. Thank you so much.
Okay. Welcome, sir. Hello.
Dear participants, I would like to share one thing about Dr.Jebasingh. After his PhD, he has visited Weizmann Institute of Israel for PDF work. And he has done a lot of work and published papers in reputed journals, and even Dr. Gopal and Jebasingh. So both of them published papers in a journal with high impact factor, that means more than 5.5. And Dr. Jebasingh is having research projects in his hand, and one project worth of less than 50 lakhs. So this is for your kind information.
Sir, someone has.
Okay.
Sir,
Someone has question in.
No, okay. So here I got the thing. So how it's being used in nanotechnology. So in the nanotechnology, what they do, one of the important function is to display the epitope. See, for example, even I also worked in the thing. I got the publication also. So what we do, like for this, you can use it as a edible, since this is a plant virus. So what happens if you want to develop a vaccine for any particular disease? So you can you can take HIV also. So what we do, like we take the epitope which is responsible for causing the disease. Okay, so what we do, we try to display in the tobacco mosaic virus also. So that will make to express in the plant. So ultimately, what happens? The particular epitope will be the particular amino acid, the peptide which has the particular amino acid will be produced in the plant. So when you feed that one, so automatically what happens? We will develop the antibody against the protein also. You will develop the thing for the several protein, but already since we insert the epitope, which is needed for the antibody production, also the antibody against the epitope will be also produced. So when the particular pathogen, when we infect, already we have the antibody by which we can have the resistance. This is one of the thing. But now it has been used for various other purposes also. So studying how the, I cannot clearly tell the terms, like it is highly useful in the vaccine field and in the pharmaceutical field also. It has a lot of applications also. So like fabrications and even for like, I don't remember now, a lot of applications there. If you need, I can send the paper also. I have one paper about that also. What is the other question? How to eliminate the white flies effectively? So this is you can do it by spraying the pesticides, or you have to follow the proper pest control mechanisms like a biological pest management. So when you so there are chemicals I don't know the name of the chemicals to kill the white flies or something. You in the online you can easily identify. So by spraying those chemicals, you can kill the vector by which you can inhibit the transmission of the virus also. So somebody asked, but isn't time consuming? I don't know what time consuming you are asking about. I don't know.
No, about nanotechnology.
Nanotechnology. No. Now it's actually the structure is known very well. And so since the structure is available, so having the structure for the core protein for any virus is critical. So I also worked on one of the Poti virus, it's called as carbon mosaic virus. When we try to have the native structure of it, like by expressing, still we couldn't have it. So having the soluble nature of the core protein is very tedious process. In that cases, tobacco mosaic is very, very easiest. So as I told you already, we have crystal structures. So it has a lot of things like purification, and for structural determinants, very easy when you compare with any protein of the virus. So people are going for the tobacco mosaic for all these purposes. So if you don't have any other questions, maybe I can stop here so we can conclude the session. Ramakrishnan, I hope I covered whatever the life cycle, whatever thing you want to expect on a tobacco mosaic virus.
Yes.
Very, very like
Yes, Jeba, really it was a wonderful.
Okay, okay.
You covered the topic extensively
Excellent.
and exhaustively.
Okay,
So
then.
thank you so much for being with us, and hope that we will also have other type of
Sure,
seminars.
sure.
Your
Sure,
presentation.
sure. Sure.
Thank you so much, sir.
Thanks for the opportunity, Kiran. Okay, thanks. Okay.
Welcome.
Okay, bye.
Thank you, sir. We thank Dr. Jebasingh for his wonder sharing of knowledge on viruses in general, classifications in particular. Now the participants, you can post your questions and chat box also. Respected participants and resource persons, if there are no more discussions, we can have a break for lunch and meet again after afternoon session at 2 p.m.
Okay,
Yes,
thank you.
we can meet at exactly at 2 p.m. After the conclusion of the second session, the feedback form link will be posted in the chat box. You can fill it out, and the e-certificate will be mailed to your registered email ID. So please do attend without fail. So we can have a break. Thank you all.